Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 7, Plant Tissue Culture. Plant cells, tissues or organs can be grown under aseptic, controlled conditions and in suitable cases, regenerated into complete plants. This ability supports rapid multiplication, disease-free planting material, conservation, crop improvement and specialized metabolite production. But tissue culture is not simply placing a leaf on a medium. Success depends on explant identity and health, surface disinfection, nutrient and hormone conditions, developmental pathway, acclimatization and quality testing. By the end, you will be able to follow a culture from explant to regenerated plant, compare organogenesis and somatic embryogenesis, explain haploid and protoplast methods, and judge each application by its evidence, limitation, and quality controls. Our roadmap begins with totipotency, the developmental potential of a plant cell under suitable conditions. We then select and establish an explant aseptically, design a medium containing mineral nutrients, carbon, vitamins and growth regulators, and examine how auxin and cytokinin responses influence callus, roots and shoots. Next, we compare unorganized callus with cell suspension culture and follow two regeneration routes, organogenesis and somatic embryogenesis. We will organize micropropagation into initiation, shoot multiplication, rooting and acclimatization. Special techniques include anther or microspore culture for haploids and protoplast isolation and fusion. We then study meristem culture for cleaner planting material, slow growth storage and cryopreservation for germplasm, and finish with contamination, genotype dependence, somoclonal variation, authentication and genetic stability. The central question is not whether tissue remains green or forms a mass of cells. It is whether the selected living material follows a controlled developmental route and produces plants of verified identity and quality. Three layers matter. Biological competence depends on genotype, tissue age, physiological condition, and totipotent potential. Culture control includes surface treatment, asepsis, nutrients, pH, physical conditions, and growth regulators. Product validation checks rooting, survival outside culture, phenotype, health status, and genetic stability. A callus does not automatically prove totipotency, and a shoot does not yet equal a field-ready plant. Reliable plant tissue culture therefore connects cellular response to organized development, whole plant establishment, and evidence appropriate to the intended application. Totipotency is the capacity of a plant cell to regenerate a complete plant under suitable conditions. Most differentiated cells contain broadly the same genetic information, yet they express different sets of genes according to position and developmental state. Tissue culture can expose competent cells to signals that promote dedifferentiation, renewed division, and later redifferentiation into organized structures. This potential is conditional. Some genotypes, explants, and cell states regenerate readily, whereas others respond poorly or only through a particular pathway. Visible callus formation demonstrates proliferation, not successful whole plant regeneration. Evidence for expressed totipotency requires development of organized shoots and roots or embryos, followed by a viable plant. In an exam, define totipotency, state the suitable condition requirement, and connect gene expression change to regeneration without claiming that every isolated plant cell will regenerate easily. An explant is the piece of plant material used to initiate culture. Its species, genotype, organ, position, developmental age, and physiological health influence contamination and regeneration. Young meristematic tissues may be responsive, but the best choice depends on the objective. Surface disinfection aims to remove or reduce organisms carried on the exterior, while preserving viable plant cells. Excessive treatment damages tissue. Insufficient treatment leaves contaminants. Because internal microorganisms may remain, a clean-looking explant is not guaranteed to be sterile. Establishment uses sterile medium, vessels, instruments, and controlled transfer. Uninoculated controls can reveal failures in medium preparation or handling. Cultures are observed for microbial growth and tissue injury. The result must distinguish contamination control from explant survival and from later regenerative competence, three related but separate outcomes. A plant tissue culture medium supplies water, macro and micronutrients, a carbon source such as sucrose, vitamins, and other supplements appropriate to the tissue. Cultured explants may photosynthesize poorly, so an external carbon source supports growth. A gelling agent provides physical support for solid culture, while suspension culture uses liquid medium. Plant growth regulators provide developmental information, and their type and concentration may change between initiation, multiplication, rooting, and embryo maturation. Medium pH influences nutrient availability and gel behavior and is adjusted before sterilization according to the protocol. Light, temperature, and photoperiod add further environmental signals. There is no universal medium for every species and stage. A well-designed experiment varies the factor of interest, includes a suitable comparison, scores defined responses such as callus, shoots, or roots, and avoids treating tissue enlargement alone as successful regeneration. Auxins and cytokinins influence cell division, expansion, and developmental patterning. In many classical culture systems, relatively stronger auxin effects favor root formation, relatively stronger cytokinin effects favor shoot formation, and an intermediate balance may support callus proliferation. This is a useful conceptual pattern, not a universal fixed ratio. Endogenous hormones, explant source, genotype, regulator identity, concentration, exposure time, and other medium components change the response. Therefore, a hormone experiment should test a matrix of conditions and record separate outcomes: proportion responding, number of shoots or roots, time to response, and abnormal development. In a CBSE answer, use the words relative balance and state that regeneration conditions are empirically optimized. Never present one auxin to cytokinin number as a rule for all plants. Callus is an unorganized proliferating mass produced from responsive explant cells under suitable culture conditions. It can provide material for regeneration, selection, transformation, or metabolite studies, but different regions of a callus may differ genetically and physiologically. A cell suspension culture is initiated by transferring suitable friable tissue into liquid medium and agitating it so cells and small aggregates become dispersed. Suspension culture improves contact with nutrients and supports sampling or scale-up, but cell density, aeration, aggregation, and shear influence growth. Both systems require periodic subculture before nutrients are exhausted or inhibitory products accumulate. Prolonged culture and repeated passage may select variants or cause chromosome and epigenetic changes. Callus quantity is therefore not sufficient evidence of genetic uniformity or regenerative capacity. Those claims need separate tests. Organogenesis is the formation of plant organs, most importantly shoots and roots, from cultured material. In direct organogenesis, organized structures arise from the explant without a prolonged callus phase. In indirect organogenesis, the explant first forms callus and organs later differentiate from responsive regions. Direct regeneration may reduce time in unorganized culture and can lower, though not eliminate, variation. A shoot is usually multiplied, elongated, and transferred to a rooting condition before it can become a complete plantlet. The origin of structures should be verified because pre-existing buds and newly induced shoots imply different biological pathways. Regeneration frequency, number of shoots, normal morphology, and rooting success are measurable outcomes. Whole plant recovery and later stability remain the final evidence that the organogenic route produced a useful plant. Micropropagation multiplies plants rapidly through controlled culture. Stage 1 establishes a clean, responsive explant. The multiplication stage repeatedly produces shoots while limiting abnormalities and excessive callus. Shoots are then elongated and rooted either in culture or through a validated ex vitro method. The final transition is acclimatisation, also called hardening. Plantlets produced in high humidity with supplied sugar and low air movement may have a thin cuticle, poorly functioning stomata, and delicate roots. Sudden transfer to external conditions causes water loss and death. Humidity, light, and nutrient conditions are therefore adjusted gradually before nursery or field establishment. Multiplication rate alone does not define success. Contamination loss, rooting percentage, acclimatization survival, true to type identity, and cost per usable plant are essential production measures. Somatic embryogenesis is the formation of embryo-like structures from somatic rather than zygotic cells. A competent cell or tissue is induced to become embryogenic, and developing structures pass through recognizable stages under controlled media. A true somatic embryo is bipolar. It establishes both shoot and root poles rather than being simply a shoot attached to callus. Induction, proliferation, maturation, and germination may require different regulator and environmental conditions. Mature embryos can sometimes be synchronized, stored, or used in synthetic seed approaches, but conversion into vigorous plants is often the limiting step. Microscopy, morphology, and regeneration behavior help distinguish embryos from organogenic shoots. As with all culture pathways, genotype dependence, abnormal development, and genetic stability must be evaluated before large-scale use. Underculture or isolated microspore culture can redirect immature male gametophytic cells from pollen development toward embryos or shoots. Because the starting cell is haploid, regenerated plants may carry one chromosome set. Haploids expose recessive alleles directly but are often weak or sterile. Chromosome doubling can create a doubled haploid with two identical chromosome sets, producing complete homozygosity in a single generation rather than after repeated selfing. This greatly accelerates breeding and genetic analysis. However, anther walls contain diploid somatic tissue, so the origin and ploidy of regenerated plants must be verified. Response depends strongly on genotype, microspore developmental stage, pretreatment, and culture conditions. A complete answer links gametophytic origin, haploid regeneration, chromosome doubling, homozygosity, and verification. A protoplast is a plant cell whose wall has been removed, while the plasma membrane remains intact. Cellulase and pectinase preparations digest wall components, and an osmotic stabilizer prevents excessive water entry and rupture. Purified viable protoplasts can take up molecules or fuse with other protoplasts. Fusion combines cytoplasm and nuclei and may allow somatic hybridization between selected parents, including combinations difficult to obtain sexually. But fusion is only the first event. The product must regenerate a wall, divide, form callus, and ultimately regenerate a plant. Chromosome composition, organelle inheritance, and hybridity require verification because mixed populations and unstable products may occur. Low isolation survival and poor regeneration are major limitations. In an exam, write the complete sequence rather than stopping at fusion. Shoot meristems are small, actively dividing regions that may contain a lower concentration of certain systemic pathogens than surrounding tissue. Carefully excised meristems can be cultured and regenerated, sometimes with complementary treatments, to obtain candidate disease-free plants. The method is valuable for vegetatively propagated crops, where pathogens otherwise pass from one generation to the next. However, meristem culture does not guarantee removal of every pathogen. The regenerated plants must be indexed using an appropriate biological, serological, or molecular test, and clean material must be multiplied under sanitary conditions to prevent reinfection. The claim should therefore be stated as pathogen tested or free from the tested pathogen under the method used, not universally disease-free. Identity and agronomic performance also need confirmation before distribution. In vitro germplasm conservation protects valuable genotypes, especially plants that are difficult to store as conventional seed or are maintained clonally. Slow growth storage adjusts temperature, light, nutrients, or osmotic conditions to reduce growth and lengthen the interval between transfers. It is useful for medium-term access, but still requires periodic monitoring. Cryopreservation stores suitable tissues at an extremely low temperature, usually after protective preparation, so metabolic activity becomes minimal. Meristems, embryos, cell cultures, or other propagules may be used depending on the species. Survival immediately after thawing is not enough. Material must resume organized growth, regenerate plants, and retain identity and important characteristics. A conservation system also requires documentation, duplicate storage, contamination control, and a recovery plan. Plant tissue culture supports rapid clonal propagation, production of pathogen-tested stock, germplasm conservation, genetic transformation, doubled haploids, somatic hybrids, selection, and specialized metabolite production. Each application needs its own quality definition. A propagation laboratory measures multiplication, rooting, survival, and prototype performance. A conservation program measures recovery, identity, and stability. A transformation experiment must confirm the genetic change and its expression. Major limitations include contamination, labor and facility cost, genotype dependence, poor regeneration, abnormal embryos or shoots, acclimatization losses, and somaclonal variation. Variation may sometimes be useful for selection, but it is a problem when uniform clones are required. Quality control therefore includes clean stock, limited passage, careful records, health testing, morphology, ploidy or molecular markers as appropriate, and field evaluation. For a 3 or 5 mark CBSE answer, begin by defining the exact process. For micropropagation, write explant establishment, shoot multiplication, rooting, and acclimatization. For organogenesis versus somatic embryogenesis, compare origin, structure, and developmental outcome. Remember that a somatic embryo is bipolar. For haploids, connect microspore origin to haploid plants, chromosome doubling, and rapid homozygosity. For protoplast technology include wall digestion, osmotic support, fusion or uptake, wall regeneration, division, plant regeneration, and verification. When discussing hormones, say relative auxin and cytokinin effects, and note genotype dependence. For disease-free plants, include pathogen indexing. Draw labeled flowcharts, distinguish callus from an organized structure, and finish with genetic stability, contamination or acclimatization as a relevant limitation. Close your notes and rebuild the chapter. Totipotency is expressed only when a competent cell regenerates a whole plant under suitable conditions. Explant choice, asepsis, nutrients, pH, environment and growth regulators shape the response. Callus is unorganized tissue. Suspension culture disperses cells or aggregates in liquid. Organogenesis produces shoots or roots, whereas somatic embryos are bipolar structures. Micropropagation requires establishment, multiplication, rooting, and hardening. Anther or microspore culture produces haploids, and chromosome doubling creates rapid homozygosity. Protoplast technology requires wall removal, osmotic protection, fusion or uptake, and complete regeneration. Meristem culture needs pathogen testing. Germplasm storage needs recovery and identity checks. Contamination, genotype dependence, cost, acclimatization loss, and somaclonal variation limit the technology. A regenerated plant becomes a useful product only after quality is verified.